Let us learn about complement system. Complement proteins. Complement proteins are group of large thermolabile proteins found in the serum and body fluids. It was first called as alloxan, named by Bordet. Then Paul Ehrlich in 1980 coined the term complement. It is a component of innate immunity, but they activate adaptive immunity. Generally, the complements are called proenzymes or zymogens. The complement components are named with prefix letters C and numbered. C1 component is synthesized in the intestinal epithelium. C2, C3, C4, factor P, factor D are synthesized by macrophages. Factor B, C6, C8, C9 are synthesized in liver. Complement activation. The complement proteins are activated by three different pathways, which includes classical pathway, alternative pathway, and lectin pathway. Classical pathway. They are activated by antibody IgM and IgG. These antibodies, when get bound with the antigen, will activate the classical pathway. IgM antibodies, which is the first antibody class produced during immune response, are effective activators. In IgG, IgG3 is effective activator followed by IgG1 and IgG2. IgG4 do not activate the classical pathway. When there is no antigen encounter, the complement proteins in serum are inactive. This is due to the protein called C1 inhibitor. When the antigen enters, this inhibitory effect is overcome by the formation of immune complex. Firstly, the C1 component will be activated. The stimuli to activate C1 include single antigen bound molecule of IgM or paired antigen bound molecules of IgG. IgM have a pentameric structure and have two closely spaced complement activating site. IgG is a dimeric structure, thus two IgG molecules must be located close to each other to have the same effect of IgM. The complement protein binds to CH3 domain of IgM or CH2 domain of IgG. Structure of C1 complex and how it is bound with the antigen. Structure of C1. C1 component consists of a stalk and three subunits C1q, C1r, C1s. C1q consists of 6 molecules with head and tail. C1r have 2 molecules and C1s have 2 molecules. They are held together by calcium dependent bonds. Classical pathway flowchart. Activation of classical pathway. 2 or more C1q strands binding to the antibody results in the conformational change in C1r where an active proteolytic site is exposed. This site will cleave a peptide bond in C1s and they become active. Activated C1s acts on C4 and then C2. C4 is a 3-chain glycoprotein with alpha, beta, gamma chains. The pro-C4 molecule exists as single chain and it is converted into C4 by the action of plasmin. The activated C1s acts on the alpha chain of C4 to cleave off a small fragment from N-terminus of C4a and the large fragment remains as it is. The removal of C4a will cause change in shape of C4b where a thioester bond exposed between cysteine and glutamine is broken. This break will generate a reactive carbonyl group that enables C4b to bind with C1 complex. C4a, which diffused away, have anaphylatoxin activity. C2 component is a single chain glycoprotein. This C2 will bind to C4b with the help of magnesium dependent bonds to form complex called C4b2b. The C2b component of C4b2b is a protease. It activates C3 and thus it is called C3 convertase. C3 is the most important complement component and it is present in higher concentration in serum. It consists of alpha and beta chain held by two disulfide bonds. The C3 component gets activated when C3 convertase acts on the N-terminus of alpha chain to cleave off a small fragment C3a. The exposed thioester bond of C3b breaks and a reactive carbonyl group is formed to bind with the cell surfaces. C3b acts as opsonins for phagocytosis. C5 is a heterodimer with alpha and beta chain. The C5 component gets bound to C3b and the C3 convertase splits alpha chain of C5 to remove the peptide C5a. The C5b fragment binds with C6. C6 is a single polypeptide chain and it is not cleaved. It forms C5b6 complex. C7 binds to C5b6 to form C5b6-7 complex, which generates stable membrane binding site. C8 binds to the active cell membrane bound C5b6-7 complex, which undergoes conformational change and penetrates lipid bilayer of the cell. The entire C5b6-7-8 complex acts as the initiator to induce polymerization of C9 as C9 is a single chain protein. The C9 molecules come together to form tubular structure called membrane attack complex MAC. MAC is the complex C5b6-7-8-9 and N is equal to 18. MAC forms large donut shape that incorporates into the bilipid layer and the central ring forms transmembrane channel and osmotic lysis of target cell occur. Next, we'll see about the alternative pathway. This pathway do not include the complement components C1, C2 and C4. Activation of C3 component is important here. The name C3 breaks down slowly and spontaneously and small amount of C3b are generated in normal serum which enables them to bind to the nearby cell surfaces. This cell-bound C3b either may be inactive or may initiate the pathway. This depends on the affinity of C3b to a protein called factor H. This factor H affinity depends on the nature of the cell surface. When the cell surface consists of sialic acid, which is the sugar found attached to the N-terminus, the affinity of factor H to C3b is higher. When the cell surface is deficient in sialic acid, the affinity of factor H to C3b decreases. The factor H alters the alpha chain of C3b, thus it becomes more susceptible to an enzyme factor I. This factor I is serine protease which cleaves the alpha chain of C3b and destroys C3b and the pathway remains inactive. Thus, most of the mammalian cell surface glycoproteins are glycosylated and thus the factor H binds strongly and masks the C3b and do not trigger the pathway. Activation of alternative pathway. They are activated by polyanions such as bacterial lipopolysaccharides, nucleic acid, virus, foreign particles like asbestos. The binding of C3b and factor H is reduced and C3b is unmasked. Thus, C3b can bind with next component factor B through magnesium dependent bond. Here you can see the flowchart for alternative pathway. After binding, they forms a complex C3b-B and they have weak convertase activity. Factor B, which is bound to C3b, is cleaved by factor D into BA and BB. The BB is bound to the complex and forms C3b-BB, which is a potent C3 convertase. This splits C3 and generates more C3b, but it has half-life of about 5 minutes only. Thus, the factor P, that is properdin, binds to the complex to extend the half-life of 30 minutes. The complex C3b, B, B, C3b acts as C5 convertase and splits C5 into C5a and C5b. Then the components C6, C7, C8, as same in the classical pathway, binds to C5b complex to form the membrane attack complex, that is C5b6, 7, 8, 9. Then the lysis of the target cell takes place. Next, we'll see about the lectin pathway. This pathway involves the production of mannose binding proteins, that is lectin. When macrophages ingest bacteria or other foreign materials, they are stimulated to secrete interleukin-1, interleukin-6 and tumor necrosis factor. Interleukin-6 acts on hepatocytes to secrete the protein lectin. Mannose is a major component of bacterial cell wall and they are found on many bacterial cell surfaces. Here you can see the flowchart for lectin pathway. Lectin gets bound to the mannose present on the surface of pathogens such as HIV, bacteria and fungi. Hence, the lectin is said to be mannose binding lectin. This binding activates an enzyme called MASP1, that is mannose associated serine protease 1. MASP1 activates another enzyme called MASP2, that is mannose associated serine protease 2. MASP2 activates the components C4 and C2. The activated complement C4 splits into C4A and C4B. C4B get attached to the surface of pathogen. The activated C2 split into C2A and C2B. C2B binds with C4B to form C4B2B complex, which acts C3 convertase and activates C3 complement. The C3b get attached with C4b2b and it functions as C5 convertase enzyme. The C5b further bind with the components C6, C7, C8, C9 and forms the membrane attack complex C5, 6, 7, 8, 9 and the target cell lysis occur. Thank you.